interest of not wasting too much of your coffee break at 10, I guess. So this is a tutorial, and we're going to talk about today, or I'm going to talk about today, about essentially how to find causal SNPs and how to find causal genes in post-GWAS studies. And I think the slides are slightly different from last year, so if you guys have seen the videos from last year, there might be something new. So my name is Bogdan Pasaniuc. I'm an assistant professor here in pathology and human genetics, and I do a lot of computational methodology work. So if you can Google us, or you can Google my group there, and you can find lots of things there. So this is a tutorial. It's not only that I'm encouraging you guys to be interactive or to talk, but I'm going to require you to, stop me whenever you guys feel like there are questions. I'm going to pose some questions to some of you guys in the room, and so on and so forth. So some of these topics will be kind of, boring for some of you, because probably some of you have already dived a lot into this. So those guys that have heard about this before, try to work or try to speak up so we can all of us talk about them. So for the purpose of this talk today, please have this following causality model in mind. Okay, this is probably one of the simplest model that you can think about, leading from germline variation all the way to disease risk, and probably it's the ground truth for most of the GWAS risk loss or most of the risk loss that have been found through GWAS. So let me quickly guide you through it. So germline DNA or DNA variations that you inherit from your parents lead to modifiers in the chromatin level, or modifiers that essentially turn on to regulate gene expression that you can capture by looking at the chromatin space or looking at histone modification, all sorts of epigenetic marks that essentially regulate gene expression, either by, let's say, in this individual that has some mutation, let's say an A there, might have a less product of gene expression that you can measure through RNA-seq, and you have this differential abundance that correlates with the germline DNA, and that essentially leads to different risks for different diseases. And here, as an example, and I'm going to use this example throughout, because that's something that we work a lot in my lab. This is prostate cancer disease status. Okay, so but for the purpose of this mental image, there's nothing really stopping you to think about your favorite disease or your favorite trait. Okay, so essentially you have germline DNA that regulates some gene that increases or decreases risk for some disease, right? And what do we do through statistics to try to capture these links? Well, GWAS links germline DNA all the way to the disease status. That's what we do in a GWAS. In an EQTL study, we link germline DNA with abundance in the transcriptome or gene level expressions. In chromatin QTL, histone QTLs, all sorts of epigenetic EQTLs, we essentially link germline DNA variation with all this ChIP-seq measured in multiple individuals. Okay. So, and the key concept here is that all these measurements are measured across many individuals. So this is a population level variation, right? And then, because most of the thing today I'm going to talk about population level variation, you can think about the entire ENCODE and roadmap as nothing more than just one line here, meaning it's one individual, right? Or obviously they haven't done it in just one individual, but they haven't really assayed throughout the ENCODE and roadmap chromatin variation across many individuals at once. Okay, so have this mental image in your mind, and because it's morning today, we're going to start with a problem. Okay, so let's quickly work through an example problem and try to see how do we go about finding causal SNPs. Okay, so let's imagine that we already have from GWAS 10 regions that are risk regions for prostate cancer, but again, you can think about your favorite disease. Let's also assume that each region has 10 variants or 10 SNPs that have been genotyped. There's LD between them, and there's no LD across the regions. So you can, for the purpose of this talk, you can think about these regions as being on different chromosomes. Okay, so again, we're not running a GWAS here. We're assuming that the GWAS has been run, 10 risk regions have been found, and now we're following them, all these regions, trying to find the causal variation at these loci. Okay, So now we have, let's say we have researchers to do CRISPR or follow-up studies on 10 of these SNPs, okay? So we start with, let's say, 10 regions, each with 10 SNPs, so we have 100 markers. Again, we make it very, very simple. We measure, association of all these markers, and our job is to pick out 10 of these markers to do CRISPR or, functional follow-up on them, okay? So let's say that we measure them in, let's say, 100,000 case or 120,000 cases of controls, and the key concept here is that we know in advance that each of these regions has at least one causal variant in it, okay? And this will be critical for the next step, okay? So here's the results of such a simulation or such a study. Here's, let's say, region 1, region 2, all the way to 10 regions. Here on the y-axis, I'm plotting something related to, let's say, minus log 10 p-value. So the highest is the strongest associated. And you can see that, you have a bunch of them here, a bunch of them here. Some region, the effect size is small enough that although they're highly significant, so let's say that this is minus 10 log p-value of 50, so that's, grossly exceeds or greatly exceeds the GWAS significance level, 10 negative 8. It's still, as compared to this region, this is much, much weaker signal. So for those of you that have been looking at this type of data, you can think about it, for autoimmune disease, this could be some HLA, this could be some other risk factors, right? So there's a variation in the amount of signal across regions. So let's start thinking about what could be methods where we can pick 10 of these variants to follow up. And our goal is to find most of the causal variants at these loci, right? So think about how would you choose them? How would you use this type of genetic data? So essentially the strength of the association or the height here. Maybe there's LD between them. Maybe we can gather LD information so we know the correlation between all these variants from let's say 1,000 genomes as external sources. What other types of information, and essentially if we gather other information, how do we combine it with this type of data? So who wants to venture the simplest, most naive way of selecting these 10 variants? Someone? Okay.
Take the top one in each window.
Okay, great. So that's the second most naive way. So the most naive way is, or the simplest way is to just simply, take the top 10 values here, right? And so let's say essentially I'm just gonna I'm going to ignore these boundaries between regions, and I'm going to say, 1, 2, 3, 4, 5, 6, and so on and so forth. And then essentially I'm going to put a threshold somewhere here, and I'm going to select these variants. Okay? Who can tell me why is that a bad strategy or a potential bad Joel?
You might just pick 10 that are in the same window. They're all just tagging a single causal variant.
That's right. So the big problem with that strategy is that you're going to oversample some regions, while by the time you get to these regions, you already wasted all of your effort to do this functional follow-up on these regions. Okay. So again, for those of you that study other traits, you can think about for autoimmunes, you'll just keep on CRISPRing or doing functional follow-up on the HLA, for instance, right? And you won't really do anything else elsewhere. Okay. So that's probably a really bad outcome of that strategy. So now we dive to the second best strategy, or the next naive strategy, which is the one that Noah highlighted, which is take the best from each of these regions. So we take the best here, the best there, this one, and so on and so forth, right? And so it so happens, because I constructed this to be a very artificial example, that we have 10 regions, we have 10 variants to follow up. You'll just select one per region, okay? What are goods and bads about that type of strategy?
You might sample from areas that have nothing.
Right, so you might sample what do you mean by that?
Well, if one of the regions has no effect, and you're choosing the top one from that region.
Sure, but we kind of assume from the beginning that we're only doing this for regions that have an effect, okay? Maybe a different way to say that is that maybe you're unlucky and your top variant, that region is not really causal. And so, for example, let's say in this region here, or maybe in this region, maybe this guy is not really the causal one. It just, by just statistical noise, it tends to be higher than the rest. Maybe the causal variant is this guy here. and then by just taking the best here, you've lost some of the signal. Okay. Make sense? Another drawback of that strategy is that by doing this one variant per region, you don't really take into account that this is a higher effect and this is a lower effect. Okay, so you don't take into account the fact that maybe here you have enough power to distinguish between this variant and this other variant, while in this region here, maybe the difference in the signal here is small enough that you're not really confident enough that this guy is really the causal and this one isn't. Okay, make sense or not? So maybe here there's such a big gap between the first and the second variant that you're pretty sure that the first variant has to be causal to explain the signal, while the difference between this variant and this variant is actually small enough that you don't really, it could be just due to chance, right? It could be just due to stochasticity. Yes?
Would it also matter for the different sample size in each of the windows? Like if one window had only like 3 SNPs and one had like 100?
Yes, so here, yes, so by sample size you mean the number of SNPs in the region, okay? So there could be different sample size in terms of the number of individuals, okay? But for simplicity here, we assume there's the same number of individuals, but the power or the strength of this peak comes from the effect size coupled with the number of individuals. So even if you have the same number of individuals, the fact that this has a large impact on the disease risk, you see a massive signal, this one has a low impact, right? But this is not what you meant, what you said. You said something about having different number of SNPs here, which is a great insight. Yes, so if you have different number of SNPs here, that will also, kind of like play a little bit of a role. So if you have 2 SNPs, okay, in the region that you typed, essentially you're picking one more or less at random, right? Because you're picking the top there. Great. Any other information that you might think about using in addition to the genetic data? Just to cover the last question here. What do you typically do whenever you have a signal, or whenever your mentor, your PI gives you a SNP or gives you a genetic finding? What's the first thing that you did? Yeah. Right, so you start looking through other databases, like you just throw it in the genome browser at UCSC, or you just look through ENCODE, look at different types of information. Do you have any ideas how you would take that type of information and try to merge it with this type of genetic data? No?
Something is conserved, I would assume it's more likely to be causal.
Mm-hmm.
Or if something is in known enhancer or something like that.
Right, but let's say that just to make it more more of a challenge for you, let's say that this is conserved with some score. This is also conserved, but with like something, let's say a score of 50. This is conserved with a score of 40. Right, in this case, fine. This is more conserved than this one, so it kind of matches the genetic data. But let's say it will be the reverse. Let's say this is conserved with a score of 50. This is conserved with a score of 10. How do you reason about these two? Right. It's more or less ad hoc. You just look at it and then you try to make a picture of it. So what we've done so far, we've essentially kind of walked through the goals of prioritization methods, and this is different from a GWAS. Okay, and this is really what I'm trying to convey to you guys, is that as opposed in a GWAS, we're trying to reject a null, and when we do fine mapping, the null has already been rejected. We don't really care about seeing whether the SNP is associated or not. They're all associated for the purpose of this. Our goal is to model selection. Our goal is to find which of these variants explains the data, right? While knowing that these variants have been rejected under the null. So the model that we discussed is marginal association statistics. So essentially we looked at by the top p-value, and we've discussed the ups and downs of this. We might intersect the significance SNPs, which is what you suggested, with functional annotations. Or for the purpose of this talk, I'm just going to label everything else that's non-genetic as functional annotation, which could be whatever. It could be conservation, could be selection, could be let's say chromatin modifiers, could be enhancers from ENCODE, and so on and so forth, right? So, and we actually discussed about 2 approaches here. One is taking the top per region, the other one taking the top overall, and we kind of figure out the difference between them. A different class of approaches are conditioning approaches, where essentially they're designed to figure out how much signal exists at the locus, right? So essentially you take the top p-value, you condition on it, and then you re-evaluate your hypothesis, right? And then you recompute all the p-values after adjusting for the top p-value, and then you iterate, okay? And then probably the one that I'm going to focus the most, or the most state-of-the-art approaches nowadays, are probabilistic approaches, where you take this data and throw it through a model that will give you at the end a probability for every variant in your region to be causal. Okay, and then from that probability you can compute credible sets for causality. You can do all sorts of interesting things to it. And because now you have a model, that it's a probabilistic model, you can integrate all these external features like functional annotations, encode, conservation, all sorts of other information, truly in a correct model that will be well calibrated. So it gives the right weight between the association signal from the genetics together with this functional annotation data. So let's go back to our example. So this is the example that we were discussing before. These are the causal variants. Okay, so this is essentially in black are the causal variants, and then this simulation essentially I made it up such that most of the causal variants are in DNA's hypersensitivity sites. So there's 20 causal variants here in this simulation. 19 of them are DNA's hypersensitivity. These are the non-causal. So you see there's a very, very strong enrichment in functional categories that could be of interest, right? So if you would think about taking that type of data and intersecting with DNA's hypersensitivity assay, as I've done here, then you would gain a lot of power, right? So these are the causal variants. So let's see how these methods that we discussed would perform in this case. So this is taking the top and then conditioning on it, and this is what happens. So essentially in most of these regions you'll be fine. So you take the top variant and then you condition on it. So you see that you pick the right one, and then essentially the second or the third causal variant in those regions tend to come up on top, right? So by conditioning you're doing in general the right thing, although in some cases you're actually doing the bad thing. So here you're essentially your causal variant just went under the threshold. And the reason is that the conditioning analysis is, in some ways, the perfect statistical tool applied to the wrong problem. Conditioning is not really aimed to find causal variants. Conditioning is aimed to find how many signals exist there. And conditioning, that you can think about it, you don't really care which variants explain what signal, which you do care in the fine map. But in conditioning, you're not really involved in this. And just to build intuition, if you have a variant that's causal and has a lot of LD friends or a lot of other SNPs in linkage disequilibrium with it, you have much higher chance of selecting the wrong variant or the non-causal variant, and after you condition on it, the signal at the causal variant will go away. So you might end up in a situation where in your data, if you do conditioning, you don't find any causal variants, which is really something that you don't really want to do, right? So that will occur a lot when you have lower effect sizes. To account for that, the most state-of-the-art approaches have focused on essentially taking that type of conditioning approach and throw it into a full likelihood model that gives you probabilities of SNPs to be causal. And I'm not going to go through the math, but it's straightforward. So essentially, the idea here is that you introduce a vector of a binary vector that tells you what SNPs are causal and which ones aren't. So you can think about the C is a vector of size, let's say 10 for the previous example, and you write down the probability of the entire p-values at that region as a function of this or conditional on which variants are causal and which ones aren't. Right, and then you can throw some priors there. This goes away because this is a constant, and essentially you're employing the Bayes theorem to say what's the posterior probability of a SNP to be causal, given all these p-values at this region, after you account for the association statistic, and this is essentially prior information that you can use, right? And the cool part about this is that you can, under very simplistic assumptions, you can just take p-values and then convert it to probability. So you don't need any extra information. And this assumption is, if you make an assumption that there's a single causal variant in the region, then these guys showed, and it has been shown before as well, but these guys showed it in this context, that you can just take p-values and essentially 2 lines of code in R, you just convert those, you take the distribution of the normal or the Gaussian of those p-values, and you essentially just sum them up and you add them to 1, and that gives you posterior probabilities for every SNP in the region to be causal under the simplistic assumption of a single causal variant in that region. Okay, and the idea is that why does it work? It works because you can fully describe the statistics at a nearby SNP if condition on the SNP to be causal as a function of the LD. Yes.
So you assume that it's typed. Would it ever be possible to predict that there is a variant that you're missing?
Yeah, so the other big assumption here, which is what you said, is that it makes an assumption that the SNP is causal in the data, but it's also you have it present. So if you haven't typed it for some reason, let's say you didn't sequence the region or your coverage is not enough, then this fails, right? Now the second follow-up, the second part of your question was whether you can use these distribution of statistics to figure out whether the causal is in the data or not. It's hard. It's very, very hard. And it's under very, very simplistic or assumption, you might be able to do it, but in general, it's going to be very, very complicated to figure that out. So getting back to our model, this is what happens if you compute posterior probabilities of under a single causal variant. So you see now I changed the y-axis. It's not p-values anymore. So the y-axis is actual probabilities from 0 to 1. Okay. So this correctly puts all these p-values that were on different magnitudes or different regions in the same scale, right? So you see that in general, the true causal variant have high posterior probability of causality, which is what you would expect, right? So you're doing a great job. You see, at most of these regions, the causal variant has a probability close to 1, but every now and then, you have these variants here that have a probability of equal to 0 or close to 0. And the problem here is that if you're making an assumption that there is a single causal variant, then all these numbers will add up to 1 essentially, because the truth is that all these sum up to 1. So then, even if you have a signal here, that will artificially be pushed down towards 0, right? So you'll have truly causal variant that have epsilon posterior probabilities here, and you can see it throughout. The reason is that you have multiple causal variants at these regions, and whenever you have multiple causal variants at this region, the fact that you assume that there's a single causal variant will, deflate those posterior probabilities. So, this is limiting. That's what this is trying to say. On the other hand, you might have, let's say in this case, you might have, this is posterior probability, you're doing the right thing, but then every now and then you see essentially false positive here. So you see a variant that has a high posterior probability of causality. It's 20%, but it's not really causal, right? And let's say you might be able to add external information here, which is in this case that it's not in a DNase hypersensitivity region, to push this downwards. Okay, so let's say that you might have some external information that tells you in advance that this is the partitioning of SNPs in the human genome. So you have very few SNPs that are coding. A lot of them are DNase hypersensitivity, and a lot of them are outside of genes. But if you take some external information that, again, I'm going to hand wave how you're going to get that. But let's say you just Google it, or you just go through the literature, or you believe some of the guys that have computed this. They told you that, well, if a SNP is in a DNase hypersensitivity, it's 5 times more likely to be causal. Or if a SNP is coding, it's 13 times more likely, right? That's what this is trying to tell you. So now, if you have that external information, okay, now you can go back to your posteriors, and then you can just reweight these posterior probabilities, because now this information is actually truly a prior information, right? So now you can take these numbers, and then you can convert them quite easily into priors, and that gives you essentially, you can just pull down some of these SNPs, or you can pull up some of the other SNPs. In general, most of these priors will act on reducing the false positive, or reducing the probability of SNPs that are non-causal towards 0, and very, very rarely would actually boost up the probability of SNPs that are truly causal. And then I can dive through it. So essentially, your taking these SNPs and you're overlapping with some functional category. But one of the big punchline here is that if you're just overlapping them, you don't really know what functional categories to do. So in this trivial example here, I gave you all this DNA's hypersensitivity and its enrichment, but in practice, you don't really know what DNA's what was the functional category that I should use for my disease of interest. And furthermore, if I use that functional category, what's the actual level of enrichment that I should use, right? So, yes, no.
Is there a way to go from enrichment to probability of S conditional on C?
Probability of S condition. Yeah, so the enrichment goes into probability of S condition. No, so the enrichment goes here. So the enrichment changes your priors on the causality status. So it goes in this part here. Doesn't change this.
So if you're 5 times enriched, is there a difference between having sort of larger effect sizes versus having more causal variance?
Yeah, that's a great question. So here, the enrichment that I'm talking about, and I'm going to get back to it. So you can think about 2 types of enrichment. One is the actual number of causal variants being larger in a category than in another. And if you just look at the count of causal variants, then that will fall in here, right? If you can think about external information as telling you not that there's more causal variants here than there, but their average effect size is larger there than in this other category, that could fall in here, right? But I haven't seen people do that. So you'll have, that could be interesting to explore. Yeah. All right, so now that we went through some of the complications here, we're going to dive through some of the methods that have been recently proposed to solve some of these problems, right? So the first one is how to model multiple causal variants, and essentially this was introduced here by an Elazar's asking group. The idea is that now the C is a causal vector as opposed to a single variant. So then when you compute these probabilities here, now you loop over all the search space of causal vectors indicators, and so it's nothing really too complicated here. And essentially the punchline here is that you can write down the entire vector or the distribution of the entire vector of the association statistics of the effect size that you see on a GWAS under a Gaussian model, where the parameters of this Gaussian model is the correlation structure of the region times here would be essentially the effect size at the causals time an indicator vector telling you which ones are causals and which ones aren't. So this would select those variants. And this would be a place where what Noah is saying could be extended. So if I'm understanding correctly, what you're suggesting would be something that, under this model, you have different effect sizes pertaining to different categories, right? So you can expand this to have effect size that's specific to different functional categories, and then you'll have to think about how to estimate those and how to actually set them, right? And so this is under the assumption that you know this. Most recent works, including these guys, have expanded this to incorporate this as into a full Bayesian model where you integrate all the effect sizes and it works better and so on and so forth, right? But the key insight here, which has started a whole wave of methods like this, is that here you can the key insight is to introduce this causal vector indicator, which sounds trivial now in retrospect, but this is really what started this whole field. The idea that now you can add an extra layer that tells you which of the variants are causal, and then you can, through that, you can impose any types of sparsity that you want. Okay, so I'm not going to dive through it. Here's some simulation result telling you that here is the probability of finding the causal variants in simulations, right? And then essentially under this method called caviar, you get more causal variants, or you find more causal variants as the number of SNPs is selected as compared to the other methods. And so this is the Mahler et al. So this is the method that assumes a single causal variant. This is the black line. This is the simple model that just you rank order all of your SNP based on p-value, and this is the conditional method. So you see that in the conditional method, you might end up, if you just happen to condition on the wrong variant, then by selecting additional SNP, you just don't improve anything, right? So you might be really under a really bad regime. Although if you just select the best variant, then you're probably in a good space. And as you increase the number of causal variants, this increases and so on and so forth. So The second type of approach is that we're trying to figure out is how do you mine this ENCODE and roadmap catalog to try to find which of the functional annotations are of interest. And I warn you that this is a massive problem. This is essentially ripe for overfitting, because, and this is this is a statement that the ENCODE consortium got a lot of slack for it, but I'm still going to name it here. So in their paper in 2012, they mentioned that by doing all this functional annotation of the human genome, so they did a lot of biochemical assays to map regions in the genome that are biochemically active, and then they made this bold statement that the 80% of the human genome is biochemically annotated. Okay, and now the discussion in the field is what this biochemical function represents, right? But I'm not going to dive through it. Today I'm going to tell you that if you have 80% of the human genome mapped or annotated with some biochemical function, you can think about it in your fine mapping experiment. You're looking at 10 SNPs, and 8 of them will have some biochemical function, right? This is you can easily make up any story that you want based on any trait that you're interested in, right? Take your favorite SNP. With 80% chances, there's going to be some annotation in the ENCODE that tells you that it has some biochemical function. The big question there is that useful for your disease of interest or not, right? Right? That's kind of like the problem here. If you mapped 80% of the human genome with function, then, well, now you have a specificity problem, right? So that's what I mean by overfitting. So how do we go about doing that? And people have dived into this concept of functional enrichment. And the idea here is to simply to say, if I have a biochemically active region in the genome, and this is not just one region, one locus. This is spread throughout. You can think about it as DNA's hypersensitivity in a given tissue type. Okay. Now you can ask yourself, if variants in these regions are causally impacting my trait, okay, let's say prostate cancer, then I would expect that the GWAS signal to be overly concentrated in those regions. Okay. So then people have defined this functional enrichment as looking at the GWAS signal divided by the proportion of the genome covered by functional annotations, right? And this is a concept that people keep on using throughout, but I think it's very easily to misunderstand. So there are two notions here when people talk about functional enrichment. One is the number of, or the GWAS signal, and the other one is the area of the genome. Okay, this is what's easily overlooked whenever you read these functional enrichment papers. So the area of the genome tells you how much the functional enrichment could be like. For example, if you map DNA's hypersensitivity in most tissues, it's on the order of 15-20% of the human genome, right? So if you observe 15-20% of your GWAS hits for your disease within that region, then essentially it's not interesting, because you'd expect that by chance, right? You just, you look at 15% of the human genome and you see 50% of your GWAS hits, then your enrichment here would be 1, right? You shouldn't be that excited about it. But if you observe, let's say most of your GWAS hits to fall within that region of the genome, then you can quantify that, right? So that's this ratio that people use as functional enrichment. So initial methods just really relied on this basic idea of just counting GWAS hits. So you can find a lot of work, even before the ENCODE and roadmap, where they looked at EQTL. So these are essentially variants that are found to impact gene expression in other studies. And then you just ask, if I look at all the GWAS hits or all the hits that come from genome-wide association studies, how many of those are EQTLs and how many aren't? And these guys quantified as 61% of the GWAS hits and EQTL spanning 49%. So you can look at these two numbers and you can ask yourself, well, is this number way bigger than this? This is like you're mapping half of the human genome and then you're observing just a little bit more GWAS hits there. Then in Morano et al. Science, they reported 60% of the hits are in some sort of a linkage disequilibrium, DNA's hypersensitivity, and so on and so forth, right? The problem with this approach is twofold at least. One is, how do you quantify a GWAS hit? Remember, there's a region of LD near every hit that comes from the GWAS. So, and that region of LD is typically much larger than the biochemical assay. So you're looking at, let's say, 100 kb, which you get a region in a GWAS hit, while the biochemical assay or the DNA hypersensitivity site would actually give you 10 or 100 or 200 bases, right? So when you overlap these two quantities, it's trickier to think about, how do you count them? How do you bin them, right? So it's not really trivial to count them. And the second problem of this type of approach is that, you're putting some very strong arbitrary thresholds on significance. So think about it. If I see in my GWAS study, I see a lot of, let's say I see 3 hits at 10⁻⁹ and 100 hits at 10⁻⁷, those 3 hits of 10⁻⁹ are all in the DNA's hypersensitivity for a given tissue, while all the other 100 at 10⁻⁷ are in a different tissue. Which one should you, how do you reconcile those two statements, right? And in this GWAS hits counting, you're pretty much going to ignore anything that happens below 10⁻⁸, right? And because you're going to say that those are just noise for you, okay? So to accommodate these two, most recent methods have been doing heritability partitioning, and this is an overloaded word or overloaded concept. Should think about it as variance explained in the trait by different classes of SNPs, right? So you're just taking all the SNPs that are DNA's hypersensitivity in a given tissue, all the other SNPs that are not, and then you just put them into mixed models, into 2 variance components with a trait, and then you essentially let them compete for variance, and you just quantify those numbers, and then you divide it by the area of the genome, right? So there's a lot of catches there, but this is a tutorial. I'm happy to talk about all the details there. There's a lot of of quirks there, how to do it. But for the purpose of this, let's say that it works. You can do it based on individual level data. So you need essentially genome-wide genetic data for most individuals in your study. Or more recently with the LD score regression methods, you just take the p-values, throw it through some fancy engine of machinery, and then that will estimate the exact same quantity under lots of assumptions, okay? So the good news for you is that you can just take LD score regression based on the GWAS associations, and that gives you that functional enrichment. So here's how it looks. These are figures from the original LD score paper from Hilary Finucane et al. So here on the y-axis here, you just have this quantity that I showed you before. So this is the proportion of the SNP over the variance explained by all the SNPs in this category divided by the area of the genome, okay? So then you see that from the baseline, you have this no enrichment. So you would expect, some variance, or if they're randomly thrown at the genome, to fall somewhere here, right? And then if different categories here denote, or different colors here are different diseases, or different traits, height, BMI, and so on and so forth. So here's the coding SNPs. They're 1% in the genome, but they explain roughly around 8-9% of the variance in these traits, right? And from this height of this bar, you can conclude that a random variant that it's in a coding region tends to be, 8-9 times more likely to explain, or explains 8-9 times more variance in traits than a random SNP in the genome. That's how you would interpret this. And then you have conserved SNPs. These are 3% of the genome, and then they explain this much, 15-20% of variance in the trait, and so on and so forth, right? While if you look at acetylation marks or variance in acetylation marks, they explain just a little bit more than random SNPs. You also have SNPs that explain less. So you have, let's say, repressed SNPs that are half of the human genome as defined by these guys. Then on the average, they explain much less variance, right, than this. And this is kind of a poor plot because it's a ratio, and they should plot it in log space so you can properly see that this is probably on the same magnitude as these guys, right? But the fact that they plot it in just regular space, you have an idea that, oh, this is not so interesting, but this is probably very interesting too, from the perspective of ruling out SNPs to be causal from your phi mapping perspective. And sometimes you see this phantom 5 enhancers, and there's a lot of debate whether this is because of the model was violated or whether this is a true signal. Okay, but there's a tutorial. So here, essentially, what you would interpret it here is that a random variant or all the variants put into a variance component model explain this much times more than their area represents, right? So this is a variance and this is immune traits, okay? So sometimes you can plot them, not just ship them, but you can plot them by, you can aggregate all these functional, biochemically functional annotations by tissue, and then you can try to use these types of approaches to find the right tissue for different diseases. Okay, so here I forget what the functional annotation they looked, but they looked at, they classified based on different tissue types. So this is brain, cardiovascular, bones, liver, and so on and so forth. And then they looked at different traits, and then they looked to see. This is also the on the x-axis now is the enrichment, okay, or the functional enrichment. And here you'd see that for schizophrenia, most of the enrichment comes from CNS or brain-related tissues, right? And then you conclude that regulation at that level in brain is relevant for schizophrenia. Yes.
I guess you can talk about inflation this way, but how is this approach different than just rainbow enrichment or whatever enrichment fully computational?
So Go enrichment, it's on genes. Okay, so here I don't have any genes.
Something like that. So hyperphylatic test with just basically take the LDB block and then you basically take the test.
Yeah, so under simplistic terms, all these things you can get them by simply, and that's a good exercise for all of you, just take the average p-value in all these bins and you'll recover most of the signal. Okay, so just literally take all the SNPs in this functional category here, okay, and then just look at their average p-values. Okay, and then you'll see that the average p-value in this category is, or minus log 10 of it, it's much higher than in this category. Okay, this methodology, the LD score regression, gives you, on top of that, gives you the added concept of variance explained. Okay, which is, you have to make those assumptions, and then you get those variance explained. But this signal is strong enough that you can get it by just looking at, the average p-values, or I think you've seen it before. I don't have it here. Or you look at stratified QQ plots, okay, which would be kind of much simpler than what you're suggesting. So you just look at all the p-values in you plot QQ plots and look at all the p-values that are EQTLs, all the p-values that are in this category, and they will be inflated more, right? And there's nothing really. The LD score regression gives you the added intuition that this is variance in trait explained. That's all that it is. And I should also say that they also in LD score they can guard you against confounder due to stratification. So one of the things that they employ in their modeling is the fact that they can distinguish between polygenicity, or the average p-value due to the trait, or the average p-value increased due to stratification in the status. Again, under some assumption. So, how do we get? So let's say that now, with like all these caveats, now we've mined through the entire encode and roadmap, and now we found the functional categories of interest. How do we take them and put them into the fine mapping? Again, so I'm not going to dive through the math of it, but it's a really cool hierarchical mathematical problem. And the idea is that now you have these functional categories here. So this is essentially the partitioning of the human genome based on all these categories that you found through LD score before. Let's say 3 of them in this case. Now you have, let's say, lots of loci. Here I'm just plotting 2 of them. And then you have the LD structure and the association statistics. And again, the hidden quantities here are which of the variants are causal or non-causal. And then you can write down a mathematical model that relates this, and you can just perform inference at it. Again, I'm not going to dive into the details, but happily to show. So this is what gets out of it, okay, from these types of models. You get, this is the association statistics for let's say triglycerides in this case, one region in the genome. On here you have the minus log 10 p-value. This is the LD structure there. This is the functional categories that were used here. So this is, let's say, acetylation in liver, and then methylation in liver, and this would be the credible sets. Okay, and this is the types of things that you can get, and these are the plots that were generated with other software, and this is generated by software that Ruthie over there has been playing with, and she'll be talking about it this Friday at Recom Genetics, for those of you that are still around. Great, so this is some numbers telling you how much you can get from adding all these extra layers of information. And again, I'm not going to dive through it. These are kind of obsolete in the sense that they've been generated, maybe 2-3 years ago. Now there's zillions of methodologies that leverage these types of ideas under different types of assumptions, okay? Where you take all these pieces of information, you model essentially a credible set, or you model the causal variant indicator, and then you spit out credible sets. And I just wrap up with a vignette saying that all these methodologies, because now you have a model, now you can truly model multiple populations, and this is really the most powerful signal that you can get from finding causal variants, because under multiple populations, let's say Europeans, Asians, and West Africans, the LD structure is different. So a variant that might be tagging the causal variants in Europeans might not be tagging the causal variants in Africans, and then you can rule that out as potentially being causal, right? So let's say a variant here, let's say you have a causal variant there, this variant might tag it here, might not tag it in this population or in this other population, and that helps you a lot in in finding the causal variants. So you can model all of them, and here's another type of plot like this that shows you under some simulations. Here's what the signal, the p-value signal that you would see in Africans. This is what you see in Europeans. So you see way more essentially variants that tag it, because the average LD block is larger in Europeans, and so on and so forth. And this is what you would observe in terms of the credible sets. So this is really, the punchline of this. I guess I don't know if I still have a lot of time, but this was supposed to be like the first half of the talk, but I see we're running out of time. So this is what you get. So you get essentially take the GWAS data, you take some LD from some reference, some functional annotations, you flush them or you mine the biochemically functional catalog like the encoder roadmap through methods such as LD score, but you can do essentially all sorts of methods. LD score is just kind of like a poster child in this case, to find the functional annotations, and then you throw them into methods, such as Painter or many others, that estimate the SNPs probability of causality. Okay, so this is kind of like the tutorial on how to find causal variants. Okay, so I don't know if we still have time to find the genes, but so I think, are you guys okay with postponing coffee for 10 minutes? Great. Wow, you guys are very, or some of you are, some of you aren't. So the ones that aren't should ask the tough questions, let's put it this way. So the second part, let's find the genes, and I'm getting back to this image that we had before. So now we just looked at these, we looked at this type of data together with this data, and we ignored the gene expression, right? So now our goal is to put gene expression in the fold, try to find which of the genes are the risk genes at this risk region. So intuitive, what we want to do the following. So let's say on the black here is the association signal that comes from the GWAS, and here we overlay it with two potential genes at that region. Okay, one of them, let's say it's the blue, and the other one is the red. And now we want to come up with some statistical methodology that tells us that this peak matches much better the blue, or the red peak matches much better the black peak as compared to the blue one, right? So we want a way to properly quantify probabilistically or statistically that, in this case, we want to take the blue gene to be the most likely gene of interest at this region, okay? How do we do that? Well, in general, the EQTL studies, so we want to integrate these pieces, but in general, the EQTL studies are orders of magnitude smaller in size than any reasonably powered GWAS. Okay, so GWAS, now we're talking about hundreds of thousands. More recently, K-Biobank is 500,000 individuals, so we have a lot of individuals in GWAS. We will never really have EQTL studies of that size, simply because you might not even have the right tissue. Okay, so if you're thinking about brain disorders, you'll never really have hundreds of thousands of expression in brains, and there's a lot of other noise measurement. How do you actually measure it, and so on and so forth. So EQTLs typically are on the order of hundreds of thousands if you're really, lucky on the right tissue. And we want to integrate these pieces of information, try to figure out which is the right gene at the region. Okay. So most people, and including us, what we played a lot with is to just essentially fill in missing data, and that's the first thing that you'd imagine by looking at this type of image. You just build a statistical model that tells you how to predict gene expression, and then you just impute it into the rest. So now you have this, essentially you have imputed gene expression or genetically predicted gene expression. You see that notion too, and then you just associate that to the disease status. The idea is that now you're testing every gene instead of every SNP. So instead of having a 10⁻⁸ threshold for, which comes from a million hypotheses that you need to multiply correct for, now you might have 10,000 if you predict 10,000 genes, but in general it's orders of magnitude. But most importantly, now what you have a gene as a unit of association. So now you truly have, okay, I'm associating this gene, and this gene is truly associated with a trait, and therefore they're more interpretable, or biologists like it. Let's put it this way. But if your SNP don't impact the trait through that abundance of gene expression in that right tissue that you measured, then you're not going to find it. Okay, so this is not a replacement for GWAS. And more recently, there's a bunch of papers that are coming out, or there's a bunch of papers that, gene expression is nothing really, from the statistical perspective, nothing really original or nothing really particular in this case. So you can do the exact same things with, chromatin variation, methylation, metabolome, any types of things that you can predict with some reasonable amount of accuracy from the genetics. Okay, and all these things will apply by just replacing to the right term. And this is showing that this has been taken over, and so there were like 3 papers in more or less in the same time last year, looking at how to predict gene expression. This is from Nancy Cox and Hakim. So this is the predict scan. This is our paper with the TWAS, and this is essentially tying this. So we extended it to look at summary statistics, and here this is tying it into thinking about these methodologies as a method for Mendelian randomization. Okay, and we're going to take that type of approach and try to understand it. Okay, so what is Mendelian randomization? Mendelian randomization is a way to assign causality under some very, very strong assumptions. Okay, so the assumptions are the following. Let's say the genotypes are your instrumental variable, so you're assuming that the genotypes have been randomly assigned to you at birth. Okay, and then the exposure is the one that you want to assess its impact on the outcome. Okay, so here exposure is gene expression. Okay, and the outcome is complex rate. Okay, and you're making the assumption that there's no confounder that impacts all 3 quantities here. So the confounder could be stratification, or could be any types of other things that you don't really measure, and you're making the assumption that there's no impact on the instrument. Right? This is where the randomization comes. This is where the assumption is. So the assumption is that your genotypes have been assigned at birth randomly. Right? If they would be assigned at birth by stratification, let's say because you're Northern European, you're more likely to have these types of alleles than Southern European, this would be invalid under this assumption, right? So that's where, and this is just stratification. It could be any other types of confounder. Okay, so there's assumption there that you can digest it, and that's a reasonable assumption. It's not going to be violated that often. Okay, if you're doing the right thing. The other assumption is that the instrumental variable, or the genotypes, don't impact your outcome, but through the gene expression. Okay, so essentially, if you think about standard Mendelian randomization, you need to make an assumption that all the effect of the genotypes on the outcome is mediated through exposure. Okay, and this is something that the more you think about it in terms of gene expression, the more you realize that it's a really, really strong assumption. So you're making the assumption that there's no pleiotropy. Right? So you're making the assumption that if the genotypes impact the complex rate, so there's an association that comes from the GWAS, all of it is mediated through expression. Okay, make sense? Yes, no? Who can give me an example where this is violated? If you think about gene expression, or an easy example where this is violated. Someone, not Noah. So what happens in my example before if I have 2 genes at the same locus? The red and the blue one. So if this is the red gene that I'm trying to assess whether that's causally impacting the trait, and I have another blue gene here, and then I say that this genotype impacts the other blue gene, which impacts the trait, okay, then I just violated the entire model. So, and you cannot think about TWAS or predict scan or SMR as a truly a Mendelian randomization. It's false. Okay, so essentially, all these slides are trying to tell you that whenever you're running imputation-based association or based on gene expression, only rarely this is truly a Mendelian randomization, or it doesn't violate the assumption of Mendelian randomization. Okay, so you should really be careful when you do gene expression imputation as thinking about those genes to be causal. It's very rare that those genes are causal, simply because most of the times you have this assumption is violated in the Mendelian randomization. Yes.
Do TWAS include each gene independently?
Yes. Yes. So if you think about the imputation procedure, what the imputation procedure is just estimating the strength of this edge under this assumption. So what you're doing through imputation, you're just essentially doing a conditional approach from a Mendelian randomization. You're just taking these genotypes, you're predicting this quantity here, and you're predicting just the quantity that's coming through this arrow, and then you're associating to the complex search, so then you're essentially predicting the strength of this arrow if you're assuming that there's no edge here. So under Mendelian randomization standard framework, by doing TWAS, you're actually doing Mendelian randomization. You're predicting this quantity here. Okay. So there's a lot of data out there. Here's a server where you can just, there's a tool that we created together with the first author of the TWAS paper, Sasha Gusev, who's going to be giving a talk here on Friday or Thursday. So you can just go to this website, and essentially you have all these EQTL weights that are pre-computed for you. So you have all the GTACs, all the brain data from Common Mind, a bunch of other EQTL weights. You can just take them and essentially just plug and play. There's a software there. You take your GWAS, and then you just compute all those TWAS and all those association statistics. Okay, and essentially now I think it's the largest catalog of EQTL weights that I'm aware of. So here's some example of how that type of model will look like. Here's a work that has been done by Nick Mancuso, who's hiding over there, in prostate cancer, 140,000 cases and controls. So this is a TWAS, or transcriptome-wide association study based on imputation, in the latest, greatest prostate cancer data, and found 201 genes association. Okay. Now the point that I'm trying to make to this slide is that every dot here is not a SNP. Okay. Every dot here is a gene. Okay. So now you have, the threshold that you cannot really see it, but it's not 10^-8. It's slightly lower than that. But you see here all these peaks that are indicative of the same Manhattan plot. Okay. So it's very hard to imagine that all these genes here at this region are actually causal. There's one gene that is causal, or maybe one or two, but all these other genes here are just tagging that gene. Okay, so you need to do fine mapping just like would you do for a regular GWAS. Okay, that's kind of like what I'm trying to show you to this plot. Yes.
Do you include non-coding RNAs as well, or?
No, I don't think non-coding RNAs are in this. But yeah, so you can do that too, as long as you have the right EQTL data that correlates genetics with those. So be wary whenever you run a TWAS, or whenever you run an imputation-based transcriptome-wide association study, that all these are truly associations. They're not really causal. Okay, and it's a leap to make the statement that they're causal. So I'm going to stop here. There are all these tools that you can use. The challenges are, if you don't have any EQTLs, then you can just boost power. Then you can do Mendelian randomization, or you can do mediation to figure out how many of the causal genes are there. So I'm going to stop here. I think we're maybe 15 minutes out of time. More questions or more concerns? Yes.
I was wondering, I may have asked this. So, have you ever thought of exploring when the signal from your GWAS disappears by doing the TWAS?
Yes. So, yes, that's right. So, I don't think it's here. Yeah. So you can essentially condition on the TWAS association and try to see whether there's any other SNPs that are associated with the trait. Okay, and that gives you some idea that that gene is actually explaining most of the signal there. And yes, we've done that, and it's quite straightforward to do.
And then it could be an indication that.
It could be an indication that.
plants
No, so it could be an indication that by having that gene, you're actually explaining the entire. Oh, you're talking about the gene at the locus or the gene elsewhere? If you're talking about the gene at the same region or the same risk region, then yes, we've done that, and then that's an indication that maybe that gene is actually truly the causal one, or it's tagging the causal gene perfectly. So then whatever the SNPs to trait association goes away. But we have never done it by doing a trans. And because there you have to worry about multiple testing or multiple genes that you can do that. But yeah, you can do all these conditionings, type of approaches to ask whether there's an extra signal after you do these associations. Yeah.
You tried using minor allele frequency as one of the annotations
When you're doing fine mapping?
yeah.
Yeah, so yeah, that's right. So you can use minor allele frequency as an annotation, but you need to be careful. So minor allele frequency is truly associated with the effect size. By using that, you can just upweight some SNPs that are more common than some SNPs that are more rare in a fine mapping framework. In general, we never really use that because it's trickier to think about how you would interpret it. But you definitely increase resolution in simulations. I don't know if that answers the question, or it's kind of it relates back to this model of whether the effect size should be inversely proportional to 1 over square root of f or 1 over f at some alpha. This LDAC versus GCT model, if that's what you're thinking about.
Well, it seems that some of these categories might also have differences in minor allele frequency of the SNPs that are within the categories.
Oh, I see what you're saying. So you're saying that, I see. So you have some sort of interaction between the frequency and the functional category. Yeah, people have observed that through LD score regression, that by stratifying on this functional categories, also on the frequency, you increase, the function enrichment under whatever some metrics. We never really used that in fine mapping, but I think that will translate to fine mapping too. Any other questions? Great. If there are no other questions, then you can grab coffee.